So if we actually look at transporting things outside of the cell, here's a SEM image so you can see what's going on with the Golgi. Lots of folds, lots of surface area. This is all membrane. So the idea is these proteins come in, get modified, and then get packaged. And when they get packaged, pieces of this membrane are actually going to pinch off. Okay? And when they pinch off and go out into the cytoplasm, they're targeted for secretion, we said. Secretion was to move something out of the cell. Now the problem is things don't just move out of the cell. Sometimes we're moving things back into the cell. We're picking up food and proteins from other cells into an individual cell. And the cell has to be somewhat protective of itself, right? Because it could be bringing in things that are somewhat dangerous. So it actually relies on these lysosomes, or we called them garbage trucks earlier, to look for anything that's dangerous, anything that needs to be broken down, anything that could be recycled. So nutrients, macromolecules that could be recycled to the cell. These are all really, really important for the cell to have in place. And they actually contain digestive enzymes that are not that much different from the enzymes inside your stomach. And the reason for that is when we bring in something like a food vacuole here, so this is just a little piece of food. Now this is not like a hunk of cheeseburger, okay? So this is going to be, food is a loose term for a large macromolecule. Being brought into the cell, the lysosome is going to attach and actually start to break down, the little purple enzymes are going to start to break down whatever the cell brought in and digest it. Now, enzyme, remember, is protein, right? Enzymes are almost always proteins. So we're going to have to build the digestive enzymes, the proteins, in the lysosomes so that we can then break down the food that is going to give us energy to make more proteins. So this becomes very convoluted, right? We also look at those lysosomes being important for breaking down parts of the cell that are no longer functioning properly. So in this case, a damaged mitochondria. The cell targets it for destruction. The lysosome comes in, breaks it down. Those pieces from the mitochondria are going to get recycled and used back in the cell again. This is very, very important. We have a limited amount of energy and resources available to our cells, so their ability to recycle nutrients, recycle whatever they can, is extremely important. Now, with vacuoles, these are essentially just large vesicles. Many folks use these terms interchangeably. The only ones that I want you to be careful with are ones that have specific functions. So, like a protist. We'll talk about what that organism is later on, but it has something known as a contractile vacuole. It's an aquatic organism, and it uses that contractile vacuole to shoot water for movement. So it essentially acts like a little jet ski. It's going to pull in water and then shoot it out to allow this organism to move. We have a central vacuole. Where did I tell you those were found? Those are in plants. And they're going to be used for water and nutrient storage. So these are very specific vacuoles, okay? So here's a picture of a contractile vacuole in a protist. So it's going to take in water and then it actually will shoot out water to help this thing move around. This is inside of a plant cell. So this contractile vacuole you see is very, very large, and its job is extremely important for water and nutrient storage. But we will also look at the fact that without that central vacuole pushing these chloroplasts to the outside, the plant would have trouble doing photosynthesis. So it's got a lot of jobs going on for it.